just finished a video on transcription. So we saw how RNA polymerase transcribed the sense strand of DNA. So we ended with this last PowerPoint, and where we talked about being able to transcribe it using this kind of shorthand. And we said this was the DNA strand. We knew it was double-stranded, and I identified the blue strand as the sense strand. Now, do note that in the cell, there's no sign that says sense strand. In the cell, the RNA polymerase can find the promoter, and the promoter is on the sense strand. That's how the RNA polymerase knows which one is the sense strand. But for our purposes, shorthand purposes, I'll need to tell you which is the sense strand. You then went and transcribed it into mRNA, and you replaced it with uracils. We have a single-stranded here, and so that's what you did. Now, I hope you still have that or recreate it, because again, we'll keep using that as we're kind of going through transcription and translation. And when I do say, okay, write this down, you will do better if you actually do it, because sometimes it's like, oh yeah, I got it. But then when it comes to actually doing it, something will come up, pen to paper, and you're like, oh, and so it'll connect it. So I encourage you to actively work with these videos. All right, so this is where we were, and I left you with the kind of teaser. Why do we do these in groups of 3? Because this is just a nucleotide sequence. There's no space in for real, but you'll see when we do the shorthand, a lot of times they're written in groups of 3. And that's because each one of these nucleotides, the 3 nucleotides, is referred to as a codon. So we have 4 nucleotides, and we have 21 amino acids that we have to encode for. So in order to do that, we can't just do 1-to-1. If I have 4 letters, I can't say every A is a phenylalanine, because then I'll only get to 4 amino acids. We can't even do it in groups of 2. AU, because if we did that, combinations of A to U, combinations of A and U, you can only, or duples with 4 nucleotides would be 4 times 4 possible combinations. So you only get 16, AU, AG, AC, AT, and then this, you only get 16 different combinations. We need to do this in groups of 3. And then we get 4 cubed, 4 times 4 times 4, and we get 64 possible combinations when we use it 4 letters in groups of 3. So that's why we do this, and this is referred to as a codon. So let's go to the next slide here. All right, so mRNA codons is what we refer to the groups of nucleotides in the 3. Each codon is going to give us 1 amino acid. So the combination AUG will always give us the amino acid methionine. It's very specific, because if we're encoding our proteins and laying down amino acids, we want to make it absolutely sure that our protein is made the same way each time. And if there's room for interpretation, it's not going to work that way. So every codon will always encode for a specific amino acid. All right, so something else I want to point out to you is that the groups of 3 on an mRNA strand is called a codon. Sometimes you'll hear the vocabulary term of a triplet and a DNA triplet. And so the DNA is also kind of written, if we go back to here, a lot of times you'll see that written in groups of 3 that correspond to these codons. They'll refer to this group of 3 in a DNA strand as a triplet. And that is just showing that that triplet is going to encode for 1 amino acid. Do not confuse the term triplet with the term gene. A triplet corresponds to a codon, which corresponds to 1 amino acid. A gene, you remember we defined gene as a segment of DNA that encodes for a protein. A protein is made up of amino acids, multiple amino acids. So a gene is going to be a bunch of triplets together that correspond to the codons, which is going to correspond to a protein. So 1 triplet, DNA triplet, is not going to encode for a protein. 1 triplet corresponds to a codon, which is going to correspond to 1 amino acid. Okay, so for some reason, every semester I get quite a few students confused about that. So make sure you know the difference between a DNA triplet and a gene, a segment of DNA that encodes for a protein versus 3 nucleotides that are going to eventually code for an amino acid. All right, so how do we figure out which of these codons is going to put in what amino acid? And for that, we need a codon chart. So this is what we call a codon chart. I will always give you this codon chart. You do not need to memorize this codon chart, but this is going to be able to tell you what amino acid comes next. Now you don't need to memorize it, but you do need to know how to read it. So that first one, if we go back to this, our first codon, and we're starting always at the 5' end. So the ribosome reads from the 5' end. Why? Because that's where the ribosome binds. It always binds to the 5' end of the mRNA. So the first one we're going to read in this direction is AUG. That's our first codon. So let's go to our codon chart and figure it out. All right, so our first nucleotide is always on this axis across the side. And so our first nucleotide is an A. Our second nucleotide is going to be across the top of our chart, and our second place was a U. So I have an A and a U, and if I follow those down, it brings me to this box here, and I get our 4 different options. It could be AUU, AUC, AUA. In our case, it's AUG. So every time the codon AUG appears, the ribosome is going to add the amino acid methionine. Okay, so this is good. Now, AUG is kind of special because not only is it going to encode for methionine, but it's also where the ribosome knows to start. Just like replication had a start and transcription, the mRNA had a promoter start. The ribosome needs a start to say, and this is your first amino acid. And that signal is AUG. So AUG is also called the start codon because not only does it add methionine, but that's also the start of all proteins is AUG. All right, so I said you didn't need to memorize any of this, but AUG, probably a good one to memorize as a start codon, just because as you're scanning down, anytime you see AUG, you know that's the very start of your protein. Okay, so good job. So we did AUG. Let's do the next one together. All right, so the next one, codon, is CUU. So let's go to the codon chart and see what we can do. I'll give you a second. All right, how'd you do? There's the C. The second spot is a U, and the third spot is a U. So we end up with CUU, and the ribosome knows to put the amino acid leucine in that place. So we have methionine, and then we're going to join it to the second amino acid, leucine, as we're building our polypeptide chain. But you'll notice that at CUU was leucine, but so CUC, so CUA, and so CUG. There is some redundancy here. Now, CUU will always give you lysine. There's no ambiguity in that. You know, the correct amino acid will always get placed in with CUU, but so will CUC. That will place down leucine as well. So there is some redundancy in this, and that's actually kind of a good thing, because remember when I talked about that spontaneous mutations like can occur when we talked about haploid, and if you get a mutation, you're more at risk. This redundancy in the DNA chain means that if your mutation occurred in this third place, so CUU became CUC, your protein is still going to form correctly. So you're kind of protected a little bit. This third position, they usually, and you'll see this happens a lot, and it's always at that third position. And they call this the wobble effect, that you can get a little bit of mutations without changing the amino acid, and therefore your protein, your phenotype, will still be the same, do the same job, and be the same thing. So that's what's going on with the third one. All right, so this is how you read your codon chart. Go ahead and go back to your strand that we've done before. Oh, I think that's the next slide, actually. Oh, no, let's do this one first. All right, so something I need to tell you in a prokaryotic cell. Many of you have a lot of information on eukaryotic cells, but this happens in a prokaryotic cell, is that, remember we talked one gene is going to give rise to one protein. And now typically in eukaryotes, you have a promoter, and then a gene, and then a terminator, and that's it. But in prokaryotes, we see a lot of these sequences where we have multiple genes on the same mRNA strand, right? This is an mRNA strand. There's our promoter. There's our terminator, and you get the same type of thing. Actually, this is the DNA. This is our mRNA strand that gets there, but the promoter that the RNA polymerase binds to is here. And then we get 3 different genes before the mRNA finishes. So we end up transcribing this long mRNA strand, but within that, we're going to have a start codon. Remember, that's where the protein starts. The ribosome puts down the first amino acid, and then we're going to have a stop codon. Translation, just like transcription, has to have a start and a stop. So the start codon, AUG, will go until it reaches a stop codon. So the ribosome has a place to bind on your mRNA. So that happens. The ribosome binds, and then the ribosome moves through. It'll start adding amino acids at the start codon, and you'll start making that protein, and it'll continue until it reaches a stop codon. And so then we get translation, and we made a polypeptide chain, which will fold into a functional protein. In this prokaryotic cell, we've got another start and another stop and another start and another stop. So multiple ribosomes can bind to the same mRNA and translate this into 3 different polypeptides. This is referred to as polygenic because we've got multiple genes on one mRNA strand, and this occurs in prokaryote cells and doesn't occur in eukaryotic cells. All right, so that's going on. And so you can see on this mRNA strand that I have lots of nucleotides that are not getting translated. There's some that are ribosome binding sites, and there's some that's just, extra. So the start and the stop is what gets translated into a protein. All right, what else do we need to say? Okay, so here's your practice. So here's your DNA strand. You're going to transcribe the red strand into mRNA. So the red strand is the sense strand. So I'll give you a chance to do that. All right, you should be able to do that because this is the same problem that we've been working on before. So you've already done that before. So now I have a strand of mRNA. We're going to start at the 5' end of the mRNA, and I'm going to put this back on your codon chart. So make sure you got it. And you now need to translate the mRNA into a polypeptide chain. Okay, so make sure you've got this down, and then I'll put it on the codon chart and give you a chance to do that. All right, how did it work? Did you get it? So you should have gotten to methionine and then glycine, leucine, cysteine, and so forth and so on. This is something you should be able to do. Use a codon chart and do that. So, okay, that's it for transcription. How'd you do? Reading a codon chart, make sure you do the practice, make sure you understand, and then we will come back with the next video and see, we've talked a little bit about translation. We're going to see at the molecular level what's happening with the structure and how that's working. Okay, thanks guys.